Okay, think about this for a second. You get a scrape, maybe a cut, maybe a small burn. What's the first thing you do? I mean, after you stop yelling.
Right. You clean it up.
You clean it up, exactly.
Yeah.
And then,
Yeah.
what's that voice in your head telling you?
Yeah.
The one passed down through, well, generations.
Let it breathe. Gotta let the air get to it.
Dry out. You want that nice, hard, crusty scab, right? Yeah. That's always been the sign, hasn't it? The visual cue that, okay, healing is happening. It looks like the body's putting on armor.
That's the folk wisdom, absolutely.
But what if that common sense, that thing that, dominated medicine for literally centuries, was actually slowing your body down? What if that scab you're waiting for is actually getting in the way of healing quickly?
That's the core question, isn't it?
It is. Okay, let's unpack this, because today we are diving deep. We're looking at a pivotal piece of science, a 1962 paper in the journal Nature by George D. Winter.
A really foundational paper.
Foundational is the word. It's short, it's elegant, and it's about research done on, get this, pig skin. And this little paper basically single-handedly overturned centuries of medical belief. It's the bedrock of modern wound care.
It really is a fascinating story, not just medically, but how science works, Our sources today, they take us through Winter's original work, but also decades of commentary showing just how crucial this was. Our mission really is to figure out how this quite short analysis proved moisture beats air for healing.
Yeah, and it's not just academic. We're talking about the spark that ignited a multi-billion-dollar industry, all based on this idea that the scab is kind of the enemy of fast healing. We're going to look at how Winter measured this cell movement with, well, frankly, shocking precision back then, and understand the basic physics, really, that makes the difference between healing in, say, 6 days versus 12.
It's a perfect example, I think, of how challenging a simple visual assumption, what you see happening with hard data, can just completely revolutionize medicine.
Totally. Okay, let's set the stage. So, to really grasp how big a deal Winter's finding was, we need to understand the world he was walking into. Before 1962, what was good wound care, according to the medical establishment?
Well, the focus was almost entirely on drying the wound out. Desiccation. That was the goal.
Desiccation, okay.
Yeah, it came from what people could see and what seemed practical, especially, before antibiotics were widespread or even in the early days. The belief was expose the wound to air, let it form that tough, dry, crusty scab. That scab was seen as vital protection.
Protection, okay. Protection from what? Just bumping it, or was there more to it, like infection?
Oh, definitely both. I mean, physically, sure, the scab was a shield against dirt, against further injury, obvious enough.
Right.
But the medical thinking behind drying it was really about infection. The logic was bacteria love warm, moist places. So if you dry the wound surface out, you're making it hostile to germs.
Okay. Create a little desert on your skin.
Sort of, yeah. And also, drying it out meant that the fluid, the stuff that naturally oozes from a wound, they exudate.
Yeah, that yellowish stuff.
Exactly. That would evaporate. Doctors back then thought that continuous weeping, that moisture, was inefficient. Maybe even a way for germs to get in or spread.
So if you were a doctor in, say, 1955, and someone suggested covering a wound with plastic wrap, trapping all that moisture.
You'd think they were mad.
Right. It would sound like malpractice. Almost. You'd be creating the perfect little greenhouse for bacteria based on the thinking of the time. Warmth, moisture, avoid at all costs.
Absolutely. The aim was stop the weeping, get a dry surface, make it sterile fast. The irony, of course, which Winter showed, is that the very fluid they were trying to get rid of, that exudate, it's packed with the body's own healing chemicals. Growth factors, immune cells, all sorts of good stuff. But they just couldn't see past the fear of infection tied to moisture. That was the mental block Winter had to break through.
And he didn't use arguments. He used data. Rigorous data. Which meant he needed the right model. An animal model that was close to human skin, but also controllable. Which brings us to the pigs.
The young domestic pig, yes. The choice was very specific, and it's part of why the paper had such impact. Pig skin, it turns out, is remarkably similar to human skin. Structurally, biochemically.
More so than, like, a mouse or a rabbit.
Oh, much more so. Things like how the collagen is organized, the number of hair follicles, the thickness of the skin layers, the epidermis and dermis. It's a really good match for us.
And the sources even mention the specifics. Pedigree large white pigs, 12 to 14 weeks old. Why nail down the breed and age like that?
It's about controlling variables. You want to remove sources of variation, right?
Yeah.
So using the same breed, same age means you're starting with pigs that are genetically similar and at the same stage of development.
Okay, makes sense.
12 weeks old means they're growing fast, so their baseline healing ability should be pretty consistent across all the test subjects. And here's another crucial bit. They were fed a standard diet, but without antibiotics.
Why no antibiotics? You'd think that might help.
Because Winter wanted to isolate the effect of the physical environment, moist versus dry. If the pigs had antibiotics in their system, it might have influenced the healing rate independently, masking or confusing the results. He wanted to see the pure, natural, biological response to the different coverings.
Got it. Pure biology in action. Okay, so we have the pigs. Now, the wounds themselves, you said these weren't just random scrapes?
No, absolutely not. These were surgically created, highly standardized wounds. They used a special instrument to make sure they were consistent, usually square, about 2 to 5 centimeters on a side.
Okay, and the depth, you mentioned that was key.
Super critical. The wounds were very superficial, only 0.01 to 0.03 centimeters deep.
That sounds tiny.
Yeah.
How deep is that, really?
Well, 0.033 centimeter is less than half a millimeter. Think about that. It's incredibly shallow.
Wow. Okay, and what layers of skin did that remove?
It was designed very precisely to take off just the top layer, the epidermis, and only the very uppermost, delicate part of the next layer down, the papillary dermis.
Papillary dermis.
Right. Crucially, it didn't go deeper into the tougher lower layer called the reticular dermis. By keeping that deeper dermal structure intact, Winter ensured he was studying the process of re-epithelialization. That's the new skin cells migrating across in its purest form. He wasn't getting bogged down by the complexities of deep tissue damage and scar formation.
Okay, so he created the perfect, shallow, uniform wound bed to watch skin regrow, like a tiny, controlled experiment stage. But how did he stop the pigs from, messing with the wounds?
Right, good point. They actually wore protective cages on their backs. These were rigid structures designed to prevent the pigs from rubbing or scratching the wounds.
Seemed like a lot of effort.
It was essential. Any accidental damage would introduce randomness, uncontrolled variables. The cage meant the only difference between the wounds being compared was the one Winter deliberately created, air exposure versus being covered up.
Which brings us right to the core comparison. He did this cleverly, right? Using the same pig for both conditions.
Exactly. A split sample design on each pig. Perfect internal control. So, the control wounds, the dry group, they were treated exactly as medical wisdom dictated, just left open to the air.
Guaranteed to dry out, guaranteed to form that classic hard scab.
Precisely. That was the baseline, representing standard practice.
And the other condition, the experimental one.
The experimental wounds, the moist or occluded group, were covered with a simple sheet of polythene film.
Polythene. Like basic plastic wrap.
Essentially, yes. A thin, inert plastic. It's occlusive, meaning it doesn't let moisture escape. Its only job was physical. Trap the natural fluid the wound produces, the exudate, and stop it from evaporating.
So it kept the wound surface constantly moist.
Permanently moist, exactly. And that simple physical difference, air versus plastic wrap, that was the variable that was about to change everything.
Okay, this is where it gets really, really good. Because Winter wasn't just looking and saying, this one looks like it's healing better. He measured it with a kind of rigor that, frankly, gives the results their power.
Absolutely. The quantification is the heart of this 1962 paper. It wasn't just observation, it was detailed histological analysis. He knew he had to measure, very precisely, the speed of epithelialization.
Epithelization, that's the new skin cells covering the wound, right?
Exactly. The rate at which those surface skin cells, the keratinocytes, migrate in from the edges of the wound and also, importantly, from the little islands of skin structures left within the wound, like hair follicles.
Oh, wow.
How fast they move to cover the raw area.
So tell us about the measurement method, because this precision is what allowed him to challenge centuries of thinking.
It was incredibly meticulous work for the time. After a set number of days, say 3 days, 5 days, whatever the time point was, they would surgically remove the entire wound area, including a bit of surrounding healthy skin.
Okay, take the whole sample out?
Yep. Then this block of tissue would be preserved, embedded, usually in wax, and sliced very thinly into sequential sections, serial sections.
Like slicing a loaf of bread, basically.
A very, very thin loaf of bread, yes. But to estimate the total area covered by new skin, they didn't analyze every single slice. That would be too much. Instead, they used a systematic sampling method. They examined every fifth section.
Every fifth slice. Why do that? Why not just grab a few random ones?
Because healing isn't always perfectly even across a wound, you know? It might be faster at one edge or slower in the middle. By taking regular sequential samples every fifth slice, you get a much more accurate picture of the average healing progress across the entire wound surface.
Avoids accidentally picking the best or worst spot.
Exactly. It reduces bias and gives a representative view.
Okay, so they have these slices. What did they do then? Look under a microscope?
Yes. They used a microscope at a magnification of times 80, so quite detailed, and they literally measured the lengths of the new epidermis that had grown across the wound bed on each of those selected sections.
How did they measure something so small?
With a special calibrated ruler built into the microscope eyepiece, called an eyepiece micrometer, the paper states the measurements were accurate to 0.01 millimeters.
0.01 millimeters. That's
Yeah.
tiny. What is that, like a tenth of a human hair?
Roughly, yeah. It's incredibly precise, measuring cellular progress at a microscopic level. And that rigor, that level of detail, is what makes the findings so compelling. It wasn't guesswork.
No room for doubt there.
Not really. And when they crunched all this data from multiple wounds, multiple pigs, multiple time points, the results weren't subtle, they were dramatic.
Okay, so what was the big conclusion? The headline finding.
The headline, confirmed again and again in the experiments, was crystal clear. Superficial wounds kept moist and occluded healed faster, much faster.
And the ones left open to the air, the dry ones?
Significantly faster, and they could put a number on it. This is the number that echoed through medicine. The moist wounds epithelialized twice as fast.
Twice.
Yes.
Double the speed. Okay, let's translate that. You mentioned the original paper had tables showing this over time. So if, under the old dry method, a wound like this might take, say, 12 days to fully cover over.
Under the simple polythene film keeping it moist, that same wound was completely epithelialized, completely covered in about 6 days.
Wow. Half the time.
Half the time. Think about what that means for a patient. Half the discomfort, half the period of vulnerability for that raw tissue. Just a dramatically shorter recovery time.
That quantitative difference, that doubling of speed, that's huge. It forces you to rethink everything. It wasn't just that moist was okay or marginally better. It was way better.
Exactly. It proved that the standard method, letting it dry and scab over, wasn't just neutral, it was actively slowing things down. It made the scab look less like a protective shield and more like, well, like a roadblock.
A break on the healing process.
Precisely. The data basically screamed, why is the scab such an impediment? What is happening at the cellular level that makes moisture so much more efficient?
Which takes us right into the mechanism, the why behind the what.
Right. So now we get into the why. And connecting this to the sort of bigger biological picture, the answer really comes down to the physics and the chemistry that skin cells need to move effectively, these epithelial cells or keratinocytes.
Okay, so this is Winter's deeper insight, isn't it? It's not just about water being present. It's about what that water allows the cells to do. Where does the new skin actually come from in these shallow wounds?
The new skin layer, the epidermis, it grows by migration. The cells creep inwards from the intact skin at the edge of the wound. But crucially, they also come from epithelial cells lining the hair follicles and the sweat gland ducts that often survive within the wound bed itself because the wound was shallow.
So little islands of surviving skin cells within the wound.
Exactly, like little regeneration centers scattered across the area. All these cells are trying to spread out, divide, and meet up in the middle to cover the gap.
So, in a perfect world, a nice moist wound bed, what does that migration look like? How did the cells move?
Ideally, it's like a smooth, coordinated sheet of cells gliding across a well-lubricated surface. The cells, keratinocytes, they extend these tiny feelers called filopodia and lamellipodia, and these grip onto the underlying tissue, the dermis, specifically the extracellular matrix, or ECM, and they sort of pull the cell body forward. They also release tiny amounts of enzymes, proteases, that can gently dissolve bits of debris or matrix in front of them, clearing a path.
So they need a smooth runway, good grip, and maybe a tiny bit of enzymatic bulldozing.
That's a good way to put it. They need a clear path, traction, and the surface they're moving over needs to be somewhat flexible and supportive. Now contrast that with the dry wound. What happens when that hard scab forms?
Right, the air exposure kicks in.
The blood and the wound fluid ooze out, they hit the air, and they dry hard. They clot and form that crust. But the real damage, mechanistically speaking, isn't just the scab on top. It's the rapid drying out, the desiccation of the layer of dermis right underneath that scab. Winter specifically pointed this out, the desiccated layer of the dermis.
Okay, so the tissue just below the scab dries out too. How does that change things for those migrating cells?
It fundamentally changes the physical nature of the surface they need to cross. That extracellular matrix in the dermis, it's mostly made of collagen and other fibers. When it's hydrated, it's relatively supple, flexible, but when it desiccates, when it loses water, it gets incredibly tough, rigid, almost like leather or cement.
Okay.
The collagen fibers stiffen, they contract, and they form this dense physical barrier.
So the nice smooth runway becomes off-road terrain.
Worse than off-road, the path of migration becomes incredibly difficult. Those epithelial cells, instead of gliding over the surface, are now forced to tunnel underneath this hard, dry, crusty scab.
Underneath it.
Yes. They have to burrow their way through the dense, fibrous bundles of that dehydrated dermal layer, and this tunneling is incredibly slow.
Why is it so slow? Just because it's harder to move through.
Exactly. It takes much more energy for the cells. They have to produce more of those path-clearing enzymes, the proteases, just to make headway. They struggle to get good traction. They struggle to maintain the right shape and polarity needed for efficient movement. Their speed just plummets.
It sounds exhausting for a cell. Like trying to crawl through thick, dry clay instead of sliding on ice.
That's a perfect analogy. The physical resistance is enormous. And it's not just the epithelial cells. That dryness also hinders other important cells, like immune cells, phagocytes, that need to move in to clean up debris or deal with any surface bacteria. So the whole process gets gummed up.
Okay, so the scab and the dry layer beneath it are a major physical obstacle. Now, flip back to the moist wound under the polythene film. It prevents drying, obviously. But is it just about the water? You mentioned the exudate earlier.
Right, and this is key. The environment under the film isn't just wet. It's filled with that natural wound exudate. And that fluid is not just water, it's biologically active. It's a soup packed with good stuff.
Like what?
Growth factors, signaling molecules called cytokines, nutrients, electrolytes. All the biochemical signals the body naturally produces to tell cells, okay, time to divide, time to move, time to heal.
Ah, so the plastic wrap does two crucial things at once.
Exactly. One, it physically keeps the underlying dermis hydrated and supple so the cells have that low-friction runway to glide across.
Lubrication, essentially.
Essentially, yes. And two, it concentrates all those beneficial biochemical growth factors right at the wound surface, bathing the cells in exactly what they need to proliferate and migrate quickly.
So it's the perfect combination. Easy physical movement plus all the chemical go signals.
Precisely. You remove the physical barrier of the desiccated tissue, and you provide the optimal biochemical environment. That combination is why you see that dramatic doubling of the healing speed.
It really highlights how the immediate physical environment of a cell is just as important as its internal programming, doesn't it? Friction matters, even at the cellular level.
It absolutely does, and it makes you rethink that natural scab formation. The body's response to injury in open air making a scab, it's probably a very effective evolutionary strategy for preventing bleeding to death or massive fluid loss in the wild, you know?
Right, a quick patch job.
A quick life-saving patch job. But in a controlled setting where we can manage bleeding and fluid loss, that ancient survival mechanism actually becomes a barrier to the fastest possible tissue repair. Winter showed us there's a better way if speed and quality of healing are the goals.
So, okay. Winter does this meticulous experiment, proves moist healing is twice as fast, figures out the mechanism. What happens next? What does this mean for actual doctors and patients? This wasn't just some tweak, was it?
No, it was a fundamental shift, a true paradigm shift, because the results were so clear, so quantitative, they couldn't be ignored. Winter's 1962 paper effectively ended the era of let it air dry as the gold standard.
It just completely flipped the script.
Completely. It replaced the old dogma with a new scientific principle. Manage the moisture. Don't aim for dryness. Aim for a controlled, moist, optimal environment for the cells.
And this wasn't just theory, right? This directly led to new products, new ways of treating wounds.
Immediately. Well, maybe not immediately in terms of products on the shelf, but the principle was established. This paper is the direct ancestor, the scientific foundation of what we now call the moist wound healing principle. And that principle underpins virtually all modern wound care.
All of it, from a small cut to something serious.
Pretty much. I mean, obviously the specific methods vary hugely, but the underlying goal of maintaining a moist wound bed applies whether you're treating a simple kitchen cut, a surgical incision, or managing really difficult chronic wounds like diabetic foot ulcers or severe burns. That thinking shift started right here. especially gathering steam in the 70s and 80s as material science caught up.
You mentioned the industry impact earlier. This basically created a whole new market, didn't it?
Oh, absolutely incalculable impact. Before winter, standard wound care was basically gauze and tape. And what does gauze do?
It absorbs moisture. It helps things dry out.
Exactly. It was designed to achieve the old goal. After winter, the goal shifted to moisture retention or management. Sometimes you want to keep moisture in, sometimes you need to handle excess moisture, but you never want that bone-dry scab situation if you can avoid it.
So that's where all those fancy bandages come from, the ones that aren't just dry pads.
That's exactly it. The whole industry of advanced wound dressings, things like transparent films, hydrocolloids, hydrogels, foams, alginates, they are all engineered solutions designed to create and maintain that optimal moist environment that Winter identified.
Let's take one example. Hydrocolloids are pretty common, right? Those sort of gummy, sticky patches.
Yeah.
How do they embody Winter's principle?
Okay, hydrocolloids are a great example. They're occlusive, so they block water evaporation like the polythene film did. But they're smarter than basic plastic wrap. They contain special polymers that can actually interact with the wound exudate.
How so?
They absorb some of the excess fluid, but as they do, they swell and form a soft, moist gel right over the wound bed. So they don't just trap moisture, they manage it, turning the exudate into this stable, gel-like layer.
So it keeps the wound moist but not soaking wet and creates a smooth surface.
Precisely. It maintains that humidity, prevents the dermis from drying out, keeps the growth factors concentrated, and provides the soft, low-friction surface perfect for those migrating epithelial cells. It's Winter's principle made into a sophisticated product.
So the industry went from simple covering to actually engineering the ideal microenvironment.
Exactly. Managing moisture carefully. Not too dry, not too wet, keeping that healing soup right where it needs to be while also protecting the wound.
And the scientific world recognized the importance of this paper, right? It wasn't just a flash in the pan.
Oh, its influence is immense and lasting. It's incredible for a paper from 1962. Our sources consistently describe it as the single most important, most foundational article in the entire field of wound care. Full stop.
Wow. And that's reflected in how often other scientists reference it.
Massively. It's one of the most cited papers in wound healing literature. The number often quoted is over 2,100 citations.
2,100 for a paper from 1962.
Yes, which is exceptionally high. It means that even today, decades later, anyone doing serious research or writing comprehensively about wound management has to go back and acknowledge Winter's work. It's the starting point. You basically can't discuss modern wound care without referencing that pigskin experiment.
It's such a powerful reminder, isn't it? We see something like a scab, it looks strong, protective. We assume it must be the best way.
Visual intuition can be misleading.
Yeah. And Winter showed that digging into the microscopic details, the physical environment at the cellular level, like simple water content, can reveal something completely counterintuitive, but far more effective.
It really underscores the power of careful, quantitative science. He needed that surgical precision, those painstaking microscopic measurements, to generate data strong enough to overturn centuries of ingrained medical habit. That's what real scientific progress looks like sometimes.
And just the elegance of the experiment itself. Using the pig model, controlling the wound depth so precisely, the simple plastic film as the variable. It's beautiful science, really.
It absolutely is. Simple question, rigorous method, revolutionary answer, #outro.
Well, this has been absolutely fascinating. Taking something we all experience, getting it cut, and understanding the science that totally changed how we heal. Let's boil it down. Quick summary, the key takeaways for everyone listening.
Okay, 3 main points. First, the old dogma was dry. For centuries, medicine believed drawing a wound out to form a hard scab was the best way to heal and prevent infection.
Right. Second, Winter's experiment proved moist heals twice as fast. His meticulous 1962 study on pigs showed, quantitatively, that keeping superficial wounds moist under an occlusive dressing doubled the speed of epithelialization compared to air drying.
Yeah, that doubling is the key number. And third, the reason is physical impedance. The dry scab and the dehydrated tissue underneath it create a tough physical barrier that slows down migrating skin cells. Moisture keeps the tissue supple and concentrates healing factors, letting cells glide easily. The scab gets in the way.
Incredible. To think that basically all of human history up until 1962, we were treating wounds in a way that actually slowed down healing, just because the scab looked like the right thing.
It's a powerful lesson. Which leads us nicely into a final thought, something for you to chew on.
Okay, let's hear it.
We learned today that scab formation, while seemingly natural, is really an evolutionary quick fix, prioritizing stopping bleeding and fluid loss over the absolute fastest tissue repair. It's a compromise.
Right. Survival first, speed second.
Exactly. So if something as basic as exposing a scrape to air significantly slows healing by creating this physical barrier, what other common medical or biological practices, things we just accept as normal, might also be based on similar evolutionary compromises? Practices that might be inadvertently slowing down the body's optimal processes simply because we're ignoring the crucial role of the immediate physical environment, things like moisture or pressure or pH or even movement.
That's a really interesting question. Are we missing other moist healing equivalents in other areas of health because we're stuck on visual cues or old assumptions about what natural means?
It makes you wonder, doesn't it? The search for truly optimal healing is always ongoing, but Winter's work shows us where one massive leap came from challenging assumptions with rigorous data.
A fantastic deep dive. Thank you for walking us through that foundational science.
My pleasure. It's a story worth telling.
Absolutely. Okay, everyone, thanks for joining us. We'll catch you on the next deep dive.